Neurophage is an innovative project that aims to photostimulate the neurons that have been denervated by degeneration. In the case of Parkinson's disease, that is our final target, the neurons are those in the basal ganglia expressing dopamine 1 receptors. And we attempt to run this photostimulation with photovoltaic nanoparticles that can be excited by infrared light. And infrared light can penetrate the tissues, so it's not invasive illumination. But we also want to bring these nanoparticles in contact with the neurons that we want to stimulate without being invasive. And to this aim, we use an innovative vector. It's called bacteriophage. It's a virus for bacteria. So it's completely harmless for humans. Actually, we have a lot of these viruses in our gut. And actually, these viruses can be genetically engineered and may also represent the new era of antibiotics. The interesting thing is that this virus can be engineered to be coupled with nanoparticles through a protein that is present in many copies. So we can get a high number of nanoparticles attached to this filamentous virus. And then it has 5 proteins on one end that can be engineered to express antibodies against the target cells. So in other words, it's a cargo that is sent and delivers the particles to the target neurons. And in this way, the particles will touch and contact the target neurons, and if they are excited by infrared light, they will excite the neurons. What we want to also to achieve is the possibility of having the virus administered peripherally, so in a non-invasive way. And we test the ability of the virus to cross the blood-brain barrier. It's a barrier that protects the brain from the external environment and toxic substances. And actually, part of the project is devoted to improve the crossing of the blood-brain barrier. That seems to occur, and the virus can cross the barrier and at the same time retain their integrity and functional activity. The final goal will be to test it in an experimental model of Parkinson's disease to see whether there is an improvement of the symptoms of the mice. Well, I think entirely, because the project could haven't been done without the support of the JPND. This gave us the possibility also to assemble a network in Europe, and our network includes, in addition to 2 Italian institutions, includes Dublin, Paris, Stockholm, and then a unit in Germany and one in the Czech Republic. So it was very nice to collaborate with these people that have a completely complementary background and activity. And actually this is an extremely multidisciplinary project that is impossible to be done in a single lab with a single expertise.